Good evening, everyone, and a very warm welcome to this very special Institute Colloquium by Professor Bruce Alberts, hosted by ISER Kolkata through our YouTube channel. So thanks to everyone who has joined us today evening. And just for the records, this talk is going to be live on YouTube right now, and it's going to be available through YouTube later in case anyone wants to catch up on it later. A request to all the members in the audience that please wait until the end of the talk to ask your questions. You can type in your questions in the YouTube chat box, and we will direct your questions to the speaker, and he will answer them as they come. So I would request our director, Professor Shorapal, to formally introduce our speaker of the day. Professor Pal.
Yeah, should we start?
Yes.
Yes. Working in the National Academy of Sciences, and he is one of the most prominent biochemists in the world. Some of the work that he has done, it touches upon various fields of biology, and he has served as the editor-in-chief for the Science magazine. And before that, he was also the editor-in-chief of the Nature for about 3 to 4 years. He is, of course, well known for his study of the protein complexes that enable replication of chromosomes during cell division in living cells. He is the author of Molecular Biology of the Cell, which is the standard cell biology textbook at most universities. I'm extremely pleased to invite Professor Bruce Alberts, who is also the Chancellor's Leadership Chair in Biochemistry and Biophysics for Science and Education in the University of California, San Francisco. So, Bruce, I'm extremely honored that you have accepted our invitation to present this Institute Colloquium. You are going to speak on science, biology, and the world's future, which is a very, very relevant topic, particularly in the present context. I invite Bruce to deliver his colloquium.
Thank you, sir. It's a pleasure to be here virtually. I've actually been at your university when it was only in one building years ago, before you built most of your buildings. And I've also visited, I spent a wonderful couple of days at the first ICER in Pune. So I really applaud India for founding these new universities. And for the students, India's future is in your hands. Science and education are going to be critical to the future of India. And that, of course, is my message, take-home lesson from this talk. But I'm going to see if I can get my slide. Now my slide's not changing. Well, that's interesting. There we go. Okay. So this is an outline of what I'm going to talk about. I'm going to talk about my own field of cell biology and how little we actually understand. Students get false information from reading my textbook and others that we understand most everything, but in fact, we have tremendous amounts of information and data about cells, but we don't understand nearly enough. So there's a huge amount of work for all you future scientists. And I also want to talk about what kind of research we need, in addition to the very trendy now trying to solve all the world's diseases. We're not going to be able to do that effectively without much better fundamental knowledge. So I'll talk about that issue, how we get our governments to appreciate that the funding of knowledge is critical. And then I will talk for students about the issue of there's so many things to do as a scientist. How do you decide what to work on, and how do you just try to distinguish something that's important and effective for you to work on from all the many things you could do? And then I'm going to end with my favorite topic. I would say that much better science education is critical for all societies, and we all have to work together on that. Even those who are research scientists have to pay a close attention to this issue if we're going to make a better world. All right, here's the start. My my information about how little we know about cells is based on writing 42 years of textbooks. I'm still working on it right now. In fact, yesterday I sent in my nucleus chapter draft for the 7th edition, but it's a great learning experience when you have to write a book, when you have to teach a course. That's when you actually struggle with deep understanding, and it makes you realize how much more there is to know. So how did I start writing a textbook? It was a complete accident. I got a phone call from Jim Watson. I was sitting in my office. I still remember it. He said, I have a new idea. I've lined up 3 other authors, and we want you to be the 4th author. And we're going to try to bring together 2 fields that were then very different. Molecular biology, which came out of chemistry and physics, and that was my field. And a field called cell biology that I knew nothing about, that really came out of microscopy, light microscopy in the 19th century, and then in addition, electron microscopy in the 20th century. And there were little there were little textbooks on cell biology, but I had actually looked at some, and then it was completely mysterious to me. You know, words like endoplasmic reticulum and parts of a cell seen by the microscope had not been really effectively attacked by molecular biology. So Watson had the right idea that this would be a good time to try to bring those 2 fields together. But the way he convinced us all to work on this book, he said, well, I've got it all organized. I got all these people trying to help us. It's only going to take 1 month next summer and another month the summer after that. And so, you know, we all signed on to try to do this, but it was enormously more difficult than Jim Watson had suggested. And I think he's got a lot of people to do important things by underestimating how hard everything is. We finally published the 1st edition 5 years later, after a total of more than a year of 12 hours days, all of the authors sitting together and working. And this is the initial set of authors. That's Jim Watson at the back. We're crossing Abbey Road in London near the recording studio that the Beatles used, because right around the corner is a little house that we've been using to write the textbook. Besides myself and Jim, the other authors all lived in the United Kingdom. So this is the last edition of the big book, and I just want to emphasize that each time we write a new edition, we're humbled by how much we still don't know. This edition, we added at the back of every chapter some of the things that are important that we don't know. There's more than 100 of these in our book, just to make sure that students don't get the wrong impression that everything there is to know is already known. So I want to now talk about my own background. I was started graduate student school in 1961. I was in a chemistry laboratory for my PhD, but it was Paul Doty who was using his knowledge of polymer chemistry to try to attack DNA and protein structures. We knew as physical chemists that molecules move very, very quickly inside the cell, you know, at microscopic scales. You know, a protein molecule tumbles something like 500,000 times per second. And we thought, therefore, that the cell was, all it had to be was a little, like a tiny test tube with a concentrated mixture of disorganized proteins and RNA molecules that were diffusing freely around and colliding, and that would make life. And of course, that was all wrong. And an early discovery that I was involved was with the importance of protein machines. We now recognize that almost every process in the cell is driven by a complex of 10 or more proteins. And these proteins work like the machines that you know about in your normal life that are driven by electricity, except that, of course, they're driven by other means. They're driven by the energy of ATP or GTP hydrolysis, most of them. That is one or more proteins in a set of 10 undergoes an allosteric transformation that's unidirectional because it's harnessed to the hydrolysis of a nucleoside triphosphate. And this gives this machine the directionality that you need to be effective. And it's just incredibly sophisticated chemistry that life possible. And here's my favorite movie, because I worked on DNA replication for 30 years, and it was a complete mystery when we started. And this is really from our textbook, but this is an example of how one machine works. There are thousands of machines, they all work differently, but this is gives you an idea about how sophisticated life is. And this movie shows DNA being replicated at the human DNA is replicated. I'll play this right now.
Using computers, we are able to picture how you're looking at an assembly line of miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called a helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
Well, that's pretty amazing process. But of course, you could repeat that over and over again for many other machines. A much more recent accomplishment was reconstituting the entire initiation process for the eukaryotic DNA replication machine. This is work done in the United Kingdom. And this is a sort of a summary of what they discovered. This is how you start replication forks and make them go often bidirectionally from a replication origin. And there's something like over 40 polypeptides here involved. But that gives you an idea of how complicated this process is. And of course, this complication is necessary for us to be able to live. But this is only perhaps a tenth of the proteins that are critical to DNA replication and DNA repair. So that we need to do the same kind of reconstruction and study of to do those things effectively. This is one example. We know that cancer starts by such a series of progressive mutations and natural selections in a clone of cells, and that's why cancer takes years to develop. And somehow you have to randomly acquire in a clone of cells 5 to 10 different mutations, and before the cancer gets very dangerous, and we call it cancer. And that's because, of course, as we evolved, complicated multicellular organisms evolved, we had to put in all these fail-safe checks and balances to prevent us all dying from cancer before we're even born. It's kind of amazing. I used to teach medical students about cancer, and I started my lecture by saying the amazing thing about cancer is that we don't all get it before we're born, because control of cell growth, given all the mutations that are unavoidable, is really an amazing, difficult feat. Anyway, this selection process for cancer has to overcome those many different blocks. And one of the early know a lot more before we can do that. So that's just an example why fundamental human problems in making humanity healthier. So an important challenge for the next generation of biochemists is doing this in vitro protein biochemistry that's needed purify all the proteins that are involved in a protein machine and study how it works for every kind of protein machine. We were surprised by how naive we were before, and here are two new things that really struck us. First of all, was the recognition that extensive sets of scaffolds, special protein and RNA molecules, produce biochemical subcompartments in the cell without requiring a membrane. Very difficult, complex chemistry that we can't understand even when we know all the parts of it, because they give rise to so-called emergent properties, and we need new ways in mathematics to try to tease those emergent properties apart. Unfortunately, I'm not going to be able to talk about that today. So this is the scaffold property proteins. This is an example from our textbook of a very simple scaffold. The idea is that when you have all these molecules that are going to interact with each other, you want to bring them closer together in the cell so that you could speed up their interactions, and also have those interactions and reactions occur in the right part of the cell to make them most useful. And so you have all these scaffold proteins and scaffold RNA molecules too, that bind reactants and make them react much more rapidly. And these scaffolds are almost always located specifically in some part of the cell, so that the reaction takes place there and makes the biochemistry much more efficient. Here's the simplest example I know about the growth of an actin filament. Actin is, of course, the major cytoskeletal filament in cells. The filament would go, all the way down through the floor for many stories in this red little molecule. It's a long filament. I'm just showing the growing plus end of the filament. And when Tom Pollard's lab studied this many years ago, he discovered that the reaction was much faster than anybody could predict, faster than so-called diffusion controlled, which meant that something must be holding the reactants next to the end. And in fact, this is what's going on. There's these scaffolds holding the actin monomer that's going to be polymerized, the red thing, into the long red filament, so that the end can grow much more rapidly. There are many more complicated ones. RNA scaffolds are also widely used, and these are the long node. Some of the non-coding RNAs work like this. And what they do is they form different biochemical factories inside the cell. This is an old micrograph of a cell nucleus where transcription is occurring. RNA synthesis is in red, and where DNA replication is occurring is in green. And these factories form when you start making DNA or RNA, and they go away when that stops. These are temporary structures, but they create these biochemical subcompartments that are very useful for the cell biochemistry. And we now realize that many of them are based on fluctuating weak interactions that we call phase separation, and they create structures called intracellular condensates. Here's just a little figure from our book about intracellular condensates. The nucleolus, a well-known structure without a membrane inside the nucleus that's been known for, maybe 150 years or more, is one such intracellular condensate. There are many others. There are probably hundreds of different intracellular condensates. In conclusion, a cell is nothing like a test tube. Nearly everything is organized inside the cell by protein and RNA scaffolds. So my fundamental conclusion is that it will probably take the rest of the century to gain a true understanding of how cells and organisms work. And we need all the students at the ICERs to help us. And it's a great challenge to do science and to make unravel some of the mysteries that remain. So my second major point is related to the first one, and that is that fundamental biological research will be essential for improving human health, as well as for progress in many other applied fields, like agriculture, for example, environment. The current problem in the United States is that our main funding agency for biological research, the National Institute of Health, has been overemphasizing so-called translational biomedical research, work directly on cancer cells, work directly on many other disease states. But with so many unknowns and so little understanding, fundamental research on biological mechanisms also remains absolutely crucial for improving human health. Not that you don't need the other one, but you need the background. And this is my favorite editorial that I commissioned when I was at Science. I commissioned 250 editorials. I wrote 50 of them myself. But this is my favorite one by Huda Zogby, who's a very distinguished translational researcher at Baylor University. And she talks about the fact that we need basic research. And she said the problem really, she had this wonderful quote that's up at the top. The trouble is that we're trying to translate a text with the sophistication and depth of Shakespeare using a first grader's vocabulary and experience. That's such a wonderful way of expressing that. Huda was born in Lebanon, so she speaks multiple languages, so she could think about that really effectively. So here's some examples of striking things that surprised many people how little we know. This paper published in 2016 was a major accomplishment, to design and synthesize a minimal bacterial genome. They use a very sophisticated synthetic chemistry to minimize the number of genes that this mycoplasm had, and they got down to 473 genes that were needed for this thing to double every 3 hours. And the amazing thing is that 149 of the 473 genes were of unknown function. So we don't even understand the simplest cell. I'm sure that there are Nobel Prizes hiding in figuring out what some of those genes do and what kind of processes are essential for life that we don't understand. But hardly anybody's working on this. It's not considered close enough to a disease problem, but it's going to be really important. Hold on, Another example is the importance of model organisms like Drosophila, the fruit fly. We're still very far from understanding how cells work together to form and maintain tissues, obviously something that's critical for understanding how cancers go wrong. And many examples show that first working on a mechanism in the fruit fly provides a shortcut to understanding humans. And as evidence for this, I take in our last edition chapter on the development of tissues has 50 references to Drosophila, which important breakthrough papers on development. This is 4 times more than the next most cited organism, which is the mouse. So, we know almost nothing about development, I would claim, if we hadn't done this work in Drosophila. Consider the human brain. This is from our textbook, but the human brain contains more than 100 billion neurons, each of which on average has to make connections with 1,000 others, according to a regular and predictable wiring plan. This is incredible complexity. And of course, out of this complexity comes human consciousness, an important emergent property. But, it's incredibly hard to think about how we're going to make sense out of this. So here's the Drosophila brain. The fruit fly's brain contains 1 million times less neurons cells than the human. And of course, in addition, you can do all this genetics, knock out any gene. You could trace every neuron, know where it is in space. And it's clear that until we understand the fly brain, we have no hope of understanding the human brain. And this understanding, of course, will guide us to. Once we have that understanding of the fly brain, it will greatly speed up our ability to deal with human brain issues like neurological diseases. And then we need new models. This is, I advertise this because it's one of my colleagues, Wallace Marshall, a young professor at UCSF, had the courage to switch fields and take up an old problem that nobody else was working on. This amazing single-cell ciliate called stentor is a huge single cell that has something like a GPS system on its surface. It knows where its mouth is supposed to be. You cut up in little pieces and put it back together again, and it reorganizes. It's just incredible. This is all this work that was done, maybe 100 to 50 years ago, and then people stopped working on it. They didn't have techniques to get any further, but he's now taking back this problem, trying to attack it with modern methods, and he deserves a lot of credit because, starting something like this is critical, but it's very hard to get support for this and to build a community that will really make a difference. And I'm sure that once we understand what's going on in this GPS system on the surface of a stentor, this will have very big applications to humans as well. And then here's our very new, unfortunately, a very new model organism, the coronavirus. This is an amazing virus. It's 4 times larger than the flu virus. It makes 29 proteins, most with functions we don't understand. Why is it so big? What are the advantages that it got from making such a big genome? The fact is, this is the most amazing mobilization of scientists around the globe focused on understanding the fundamental cell biology of the single virus. And this is going to be really important for creating many new fundamental understandings, both about viruses and about the human immune system, which many of these proteins are designed to counteract. So there's got to be some advantage to this disaster that's around us, and this is one of them. So now for students, how to decide what to investigate as a scientist. This is a big question, very important. I'll just briefly describe how we try to address this question in a course that I've been teaching for years to graduate students at my university, UCSF in San Francisco. So we have a series of some 40 different mini courses. These are courses which are taught over the course of 3 weeks in 10 3-hour sessions, 3 hours a day. And there are 15 students meeting with 2 faculty, which the students read and discuss a series of research publications. And any 2 faculty get together and propose anything they want to teach, or what, if you want to learn something, you want to teach it. And the mini course that I've been teaching is called Classical Papers in Biology. And I'm now teaching this course with Denitsa Fujimori, a young pharmaceutical chemist. And the important point is we use history in classical papers with the aim of stimulating students to think innovatively and planning their own thesis research, because these students are just about to choose a research laboratory, the first-year graduate students, and start their career in research. And we start with Mendel, and we pair every classical paper with a modern one in the same field. And so we pair this one with a modern paper about epigenetic inheritance. But basically, we try to see how these scientists did their work and why, and what was the strategy, and what made this a great contribution. We read many Nobel Prize winners' papers, and so one is the papers by John Gurdon and Shinya Yamanaka. Yamanaka, of course, developed ISPC cells and iPSC, and he has a great video about how he chose a research topic and how he devised a successful research strategy that I recommend to all students. And if you Google Yamanaka Lasker Prize, you'll find this video. We do biochemistry by starting with Arthur Kornberg's discovery of DNA polymerase and ending with single molecule assays, which are really amazing. 4 other papers taught on successive days, and we change the topics from year to year, but we always end with Darwin and scientific standards, because we want to end with giving students a push to set a high standard for themselves. And so we read 2 chapters from Darwin's famous book, Origin of the Species, and we pair it with a recent high-profile Arthur paper from famous authors that have been shown to be completely wrong. Something like 20 authors on this paper, many of them students and postdocs. And so the whole point is to try to talk about how to avoid being misled, fooling yourself as a scientist. And the reason why we pair it with Darwin is he's wonderful in trying to point out what could be wrong with what he thought and try to study whether he was wrong or not. And he has this wonderful quote from his autobiography, which is what we want all scientists to do. He said, "I had also during many years fallen the golden rule, namely, whenever a published fact, a new observation or thought came across me which was opposed to my general results, to make note of it without fail and once. For I found by experience that such facts and thoughts were far more apt to escape from the memory than favorable ones." He's trying to prevent himself from being fooled. And that's such an important thing for any scientist to do, be honest. So this is, of course, we had to teach this year by Zoom. And this is teaching about Darwin, showing him his wonderful home where he did most of his work at Down House near London. And I thought these are pictures I took myself of Darwin's desk for inspiration. And they're the students on the Zoom. So our major point is strategy is everything in science. As students discuss each paper, we dissect the experimental strategies that have been designed by each scientist to attack an important biological mystery. We point out that these often involve an investment in developing a method, such as Yamanaka's assay that allowed him to test a mixture of 20 possible reprogramming factors at one time. Doing science is like painting a great picture. That's a wonderful thing about science. Is you have a chance to be very creative. And I talk about the fact sometimes that you could give me all of the paints and materials that Rembrandt had, and I couldn't do anything useful. He had a great talent for painting a picture. Scientists too often just measure things without any strategy. It's too easy to do experiments and get data. The great art of science is to do something really important and do it in an original way, so you make a unique contribution. And so it really takes creativity, and that's why science is to me, is such a wonderful career. So I want to talk now about India, because I spent a lot of time in India. And I learned a lot from India about the power of science to improve people's lives, everybody's life, and how science is really critically important in many different ways to the world. Here's a picture of me in Pondicherry in 1998 at these information villages that were reached. Farmers' villages reached by wireless internet that were set up by the M.S. Swaminathan Foundation. And here I am with Swaminathan with a bunch of bankers who are making loans to these groups of village women so they could start little quote industries, dairies. Here's a little science franchise where the Indians were taught by the scientists using wireless internet how to make biopesticides, parasitic wasps, and they therefore created this little business that you see here selling these eggs. And then I made a wonderful long trip to India at the end of my term as editor-in-chief of Science Magazine with the Science News team, where we visited IITs and many other places, and ended up writing this wonderful, the news staff wrote the article, this wonderful article, Science for All. This issue of this magazine had a picture of an Indian woman farmer on the cover. But very admirable ways in which it was shown how science could really contribute importantly to the livelihoods of even the poorest people in India. And then the really critical thing that I learned, and this was at my first trip in India in 1993, is about Nehru's coining of the term scientific temper. And it has really changed my view of what science education needs to be like, and how important it is in society. So as Nehru knew very well, every nation needs much more of the creativity, rationality, openness, and tolerance that are inherent to science. Nehru wrote about scientific temper over and over, and this is why you have an ICER. This is why you have IITs. India made huge investments in science and technology, and these are the values of science that I'd like to emphasize. Honesty. If you're not honest, this Darwin principle. If you're not honest, you destroy science. You have to be straightforward with what you report so that other people can build on it. There's a generosity, a strong demand for evidence with openness to all ideas and opinions, irrespective of their source. Scientists has to listen to a graduate student's ideas or an undergraduate's ideas with the same attention and respect that Nobel Prize winners' ideas get, because the graduate student will maybe have the better idea. That's the only way that science can work. It depends on the idea, not on who says it. So my final bottom line is that to produce a scientific temper, there's a need for us to really do major changes or redefine what we mean even by science education. I think all countries have this problem, but this is the United States. Something that happened to me after one of the talks I gave at a university, a mother came and said her 8-year-old student had just come home from school and told his mother, who was a scientist, "Now I get it. Science is just like spelling. You need to memorize it, and it doesn't make any sense." This is a tragedy, and it gives everybody the wrong idea of what science is, and causes, I think, a lot of problems in our society with politicians and others who don't respect science. So this is the kind of science education that I think needs to be done everywhere. It's the curriculum that we actually had implemented in San Francisco 1993, when we had a wonderful leadership in the school district. Every year of school, we had really good science for the kids. Starting at 5 years old, they would do this kind of exercise, which just shows the kind of science that can be done, not memorize, but actually investigate. At the right time of year, when there's seeds on the ground, the students were given clean white socks, and they walk around the schoolyard. They come back in, the socks are dirty. They pick off all the specks and put each black speck on the sock in a different numbered square, and then they look at each speck and draw it with a little tiny little microscope, plastic microscope. And then they have an argument about, the teacher says, well, some of these are seeds because seeds evolved to stick to animal fur. Most of them are dirt. Which are seeds and which are dirt? And eventually some student will say, well, I think the regularly shaped ones might be seeds, and the ones that are not regularly shaped are dirt. And then if the teacher teaching this right, she doesn't say, yes, that's right. She says, well, what do the rest of the class think? And then eventually the class will think, well, that's a possibility. And then the next day they might discuss, how we're going to test this idea. And again, the teacher doesn't tell them that some 5-year-old kid will eventually suggest we could plant all the regular shaped ones in one pot and take all the other ones that are not regularly shaped and plant them in another pot and see if we only get plants from one of the pots. And, and eventually the class decides that's a reasonable thing to do, and they do it. So this is the kind of thinking that we need everybody to learn how to do. And this is an example of what 12-year-olds were doing in San Francisco when I left. Of course, the challenges are more difficult as you get older, but science educators have developed all these wonderful problems to solve for students at different age levels. But the point is that the students work in groups, the classroom is noisy, and the teacher is a coach. The teacher's in the back there and walks around not talking to students, lecturing at them, but helping them discover answers to problems. So let's imagine an education that includes solving hundreds of such challenges over the course of 13 years of school. I think that this would make a huge difference for society, because these people, once they're adults, would be great problem solvers in the workplace. This is the kind of thinking that every business needs. There will be people who are able to make wise judgments for their family, their community, and their nation. And they will be adults who reject what Carl Sagan talked about as magical thinking. We have too much magical thinking today in our society. United States, we've got internet spreading all kinds of crazy things about the coronavirus, trying to argue that masks cause disease rather than prevent disease, dangerous kinds of things. And we need people to be able to analyze for themselves based on evidence what's true and what's not true. Too many people are being fooled, and our society is having a terrible problem with the virus for this reason. So to remove a major barrier to progress at the lower levels before college, I would claim that science education at the college level must change. And this is something that professors don't want to hear. At the academy, when I became president, I was there for 12 years, and I spent a lot of time trying to get the academy to help with science education. But many academy members didn't think it was our job. They said this is somebody else's job. But in fact, what they don't realize is that, how you teach the first year of biology at college defines what biology science education is. And so if we teach first year college biology is all the words of biology, all the facts that biologists have to understood in a very superficial level, then that becomes a model for teaching biology at all other levels. And you end up with these horrible courses, textbooks for 12-year-olds, which have 500 words to memorize and associate with a few sentences, words like endoplasmic reticulum, a structure that I didn't learn about till I started to write my textbook. But now in the United States, in some standards, they want every 12-year-old to know what the endoplasmic reticulum is, which is ridiculous because they can't understand anything. So we're destroying science education by including huge numbers of science facts. So this is what college biology class should look like. It's what it does look like at the University of Minnesota and a few other places. The University of Minnesota has something like 3,000 students taking the first year of biology class, and they divide the class into 15 different sections of 200 students each in a room like this, and it's a noisy classroom, just like the classroom for 12-year-olds. And we're trying to push all science education in the United States to be like this, but it's a slow process. An important tool is this wonderful analysis and guide from the U.S. National Academy of Sciences. It's a free download. It's called Reaching Students, What Research Says About Effective Teaching in College, and how to change your nobody changes their teaching all at once. It says, start try something different for 1 week, and then and it tells you why and what the evidence is that this makes a huge difference. A major problem is that the people who become professors are all people who were good at learning from lectures. And we think everybody learns that way, but research says it's not true. And yeah, at any rate, if college faculty don't change, the lower levels are not going to change effectively. All right, so I just want to end, because this is an ICER, it's science education and research, with the goals for science education, very ambitious, increasing ambitious goals. The standard thing that we want, everybody has thought about for science education, is number one. Is to provide all adults with a general sense of what scientists have discovered about the world. You know, that the universe is huge, all life is made of cells, and so on. And this is, of course, important that you know something about science. You don't need to know about the endoplasmic reticulum, but you need to know the fundamental broad thing. But a more ambitious aim would be to provide all adults with an ability to investigate scientific problems as scientists do, using logic, experiments, and evidence. And this is a focus of the inquiry-based science education that I just discussed. You know, those 5-year-old kids learning how to figure out which are seeds or which are dirt. Okay, so that's been a movement that we've been working on for many years. We're making slow progress. It's the kind of thing that the college students were doing also in that picture I showed you. Then more ambitious would, and very important though, we're realizing today, is to provide all adults with an understanding of how the scientific enterprise works. What is science? How do we know that that the smoking, vaccination, and climate change are important issues? So why they should trust the consensus judgments of science on, and I think they only going to do that if they understand how science works. And we need a much stronger focus on this goal, and we need to investigate how to do that most effectively. It's going to take education and research, and much more focus on that. And then finally, the really ambitious thing is to provide all adults with the habit of solving their everyday problems as scientists do, doing logic, using logic, experiment, and insisting on evidence. And this is critical for all our societies, to especially every democracy. And this will require a transference for what is learned in science class to general habits of mind. And again, we need experiments and to learn how best to do this. But all this says that science education needs a much larger place in our education systems. And none of this is going to happen without the involvement of scientists. It's just scientists have to lead the way to help teachers and support teachers to do new kinds of science teaching. And again, we have to start by doing all these things at the college level, so that the people who become teachers will teach the way they were taught, and redefine what we mean by science education. So this is some examples of what can be done. There's a new project driven by the World Science Academies called Science for Global Goals. And what it is, now there's already 3 curricula available on the web. There's one on the virus, and one about food, one about mosquitoes. And this is community-based learning. And it involves volunteer scientists and others helping kids actually investigate their own world. This is some examples from the mosquito unit. That was the one that was first developed. But all these things can be found as free resources on the web, and the plan is to develop all these different modules on energy, water, pollution, so on and so on. Those of you like to know more about it, just go to the Smithsonian website. The leader of this project is Dr. Carol O'Donnell, who's the director of the Smithsonian Science Education Center in Washington, D.C. And you could contact her if you want to know more. But I think it's a great resource that needs to be spread by scientists everywhere. And this is my favorite quote about scientific values. You know, science is so much more important than most scientists think, because we really haven't had time, most of us, to look beyond our own research. But Jacob Bernowski wrote this wonderful book called Science and Human Values. And he wrote it after flying over Hiroshima and Nagasaki as a young man in the British Army and being disillusioned about what the atomic weapons had done and wondering whether science was good or bad for the world. And he concluded that science was, in fact, very good. He said the society of scientists is simple because it has a directing purpose to explore the truth. Nevertheless, it has to solve the problem of every society, which is to find a compromise between the individual and the group. It must encourage the single scientist to be independent and the body of scientists to be tolerant. Science has humanized our values. Men and women have asked for freedom, justice, and respect, precisely as the scientific spirit has spread among them. Just really a critical thing to keep in mind. And this is the promise of world science working together. You know, I spent a lot of time with, when I was Academy president, going around the world and meeting with the presidents of other science academies, and even made a visit to Iran where our governments were not talking to each other. Scientists everywhere share a common culture, and we are all unhappy with our governments making short-term decisions. This is taken at a Swiss McDonald's where there are 15 Academy presidents present, but I didn't get them all in the picture. These are presidents, two Indian Academy presidents, Prakash Tandon and Govardhan Mehta, close friends of mine, and then the president of the Japanese, Chinese, and Swedish academies. So basically, science could be a great force to bring people together across the world. The problem with this group is that we're not on social media. And the world's on social media. So we need a new kind of organization called the Young Academies. The Germans deserve a lot of credit for inventing the Young Academies. And these are pictures of the two of the early Young Academies, South Africa and I guess it's Sweden. I can't see it. And how do these academies differ from traditional academies? First of all, election is temporary for 5 years old only. In typically, they're future leaders, age 35 to 40, and then of course you graduate out of that. Otherwise, it wouldn't be a young academy. But very importantly, accepting membership requires an obligation to spend time on public service. And there's at least one major Young Academy project each year directed by an executive committee of members. And finally, membership is broader than a typical science academy usually, including not only engineers and social scientists, but in many academies also political scientists, science communication experts. The first Young Academy I visited was in the Netherlands, and the president that year was a woman lawyer to connect science to other parts of society. And here's your founding members. You may recognize somebody in here. Of the Indian Young Academy, and the Indian Young Academy now has 96 members, and it's something you should all know about. And hopefully they're going to make a big difference for India. And this is my last slide. You know, to create a scientific temper for the world, there are many challenges ahead for scientists, but by working together across the globe, we shall find a way. This is my last visit to India in Chennai at M.S. Swaminathan's Institute, and here we are working with school children on hands-on science. So thank you very much.
Thank you for the excellent presentation, Bruce. Are you ready to take on a few questions from our members of the audience?
Sure.
Okay, so we have a question from Sharanya Chaudhary. So she is asking, Rosalind Franklin's image 51 clearly revealed the shape of the DNA. Why is she not credited for that?
She,
Controversial question to begin with.
well, she died before the Nobel Prize was awarded. So that's one reason. There's a lot of stories that I mean, it's been written about very well many times. Aaron Klug, who was a Nobel Prize winner himself and a student of Rosalind Franklin's, wrote a wonderful article about her contributions, and I think was published in Nature. So if you Google Aaron Klug, Rosalind Franklin, you'll find that it's a long story, and it's been analyzed to death. But there is a problem with, there has been a problem of acceptance of women in science. We're making progress. I hope. I think there's been a huge change over my lifetime in the number of prominent leaders of American scientists who are women. The president of our academy is now a woman, Marcia McNutt. If a country is going to be successful in science, we have to make maximum use of all our talents. And that if you're not making maximum use of female talent, you're losing half your talent. So it's a very active issue. In the United States, we're now focused, in addition, on bringing more of minorities into science as leaders, because half of our population now are not. Very soon, half of our population will be people who are not white, and yet the leadership in science is predominantly white. So we're really actively working now, focusing on bringing in Hispanic and Black leadership in science.
We have a more corona-focused question. So somebody is asking, would the fixation on the coronavirus research affect other fields of research in science?
I hope so. I hope so. Well, right now, it's setting a new example for cooperation among scientists. People are working by Zoom, and Zoom has been a great, what we're on now. Zoom has been a great experience in the sense that we've all learned how to do this, and we realize that we don't have to travel everywhere to give seminars. I've been giving, I gave a talk earlier this week to Portugal. So, but more broadly for science, I would like to see new experiments done and getting people to think together and cooperate using Zoom and other features, and regular meetings by Zoom to brainstorm about what are the important things, unsolved issues. I'm just finished writing drafting the chapter on the nucleus for our next edition of the textbook, and it makes you realize how little we know about important things, and very importantly, how everybody goes their own little way and does their own little bit in measuring things. But we need a community to come together and say, what are the important things we need to do to really make progress here? I could give you a whole list of those now, since I just wrote the chapter. Some of them are hard, and so nobody does them. But by cooperating, we could get, if we had got people to cooperate together more effectively, we could make much more rapid advance in science. You don't. I think we're going to try some experiments. You don't need to be co-publishing with somebody to collaborate intellectually to try to develop the field. You don't have to always publish with them, but you should be thinking with them. And so I hope we could do better in science as a result of this experience, for both because of our realization that we could do remote cooperation. That we used to think if you're going to have a meeting by Zoom, or a virtual meeting, that was like not an important meeting. It was just a, you know, a 1% solution to whatever you're doing. Now I think we could do 50% of what we do at a regular meeting. So I think there will be benefits coming out of this. I hope.
This is interesting because I think the person who asked the question was actually thinking of the negative aspect of too much corona research, but you actually came up with a positive answer to that question. So that's really nice. Uh, Another question we have is, how do you highlight the importance of basic research over translational research to the layperson?
Well, my academy actually produced a bunch of useful documents. They're still on the web. That was when I was there in the 1990s to explain to our government about basic research. It's called Beyond Discovery, the Path from Research to Public Benefit, to Human Benefit. And in each example, we took something that was really important for humanity. One of the things we did was a cure for AIDS, cure for HIV/AIDS. And we traced back where the knowledge for that came from. And we produced a series of 20 8-page pamphlets. They were designed for the public and for our politicians. And I think they were very effective. They showed the first one we did was on the global positioning system. And we showed how that started with physicists discovering atomic clocks. And it took 30 years, and then we have a centerfold in each one of these pamphlets, which traces how a piece of evidence was like atomic clocks was combined with something else and with something else, and over 30 years, eventually, in ways you could never predict, you had this amazing breakthrough where you have 24 satellites going around the Earth with atomic clocks in them, and how long it takes for that satellite to get to you tells you where you are. And we did that over and over again. And I think the scientists who worked on those learned a lot about the answer to that question, how's basic science. In fact, I'm convinced that the most important innovative things that have been developed for humanity have come out of unexpected ways of knowing how the world works, like the atomic clock, those tools. So those pamphlets are a good example. I think every society should, India should develop that develop some of those things for your politicians and for your public. The fact is that it doesn't give you immediate payoff. It takes decades before fundamental understanding leads to enormous benefit. But we need these investments. So the only way to really explain that, I think, is to go back in time and show how it's been true in the past. And it's going to continue to be true in the future, as I emphasized in my talk.
I think I'll take a couple of more questions and then we can wrap up. We have a lot of comments coming here, people praising and thanking you for the wonderful talk. So we have a question from Professor Tapush Sengupta, who is a faculty in our Biological Sciences Department at IISER Kolkata. He's asking, should publications be mandatory for students to submit PhD thesis?
To get a PhD, you mean for the PhD? Yeah. At least I think the new way of thinking about it is a paper in bioarchive. Yes, you shouldn't have to wait till it's published. So my colleague Ron Vale at UCSF was responsible for initiating this new tradition of putting your papers in bioarchive or in some kind of preprint server. And part of the reason was that students were waiting way too long to get their paper published and was holding up their PhD. But I think you need I think for a PhD, you should be required to develop enough research that it becomes the substance for a paper, but you shouldn't have to wait till it's absolutely published, because sometimes it takes years going back and forth. You could be, your PhD could be evaluated from the preprint. Your committee could look at the preprint.
Thank you. One last question, and then we'll wrap up the session. Otherwise, it's getting quite late for all of us. This is again, you can choose whether you would like to address this question or not. Somebody is asking, would you comment on the relationship between science and spirituality?
Yeah, this is a very important question. My academy has spent a lot of time on this. In the United States, there's been a conflict in biology education between people who want evolution taught as one is an alternative theory to other interpretations like divine creation and in something that was called intelligent design. And scientists, including the academy, are insisting that science cannot be mixed with religion, that in science class you have to teach evolution because we know that that's what happened. Darwin started that, but there's huge amounts of evidence. And all of biology depends on understanding evolution, including how cancer by an evolutionary process. And at the same time, our academy has published these series of booklets, which you can find on the same academy website, called Science, Evolution, and Creationism, which argue that science should not interfere with religion. We have no basis for evaluating religion. It deals with a different aspect of human existence. It's not something you could do experiments on, get evidence on. And our little pamphlets have statements at the end of them by famous bishops and religious leaders agreeing with us that those two domains must be left separate. And so I would refer the people interested in this to go to the Academy, National Academy Press. It's called National. If you Google National Academy Press, you'll find all these. There's actually 10,000 books online for free as PDFs. And if you search that site for creationism or science and evolution, you'll find lots of these little pamphlets which are designed for the public, but they're very clear in making the distinction between science and religion and treating both with respect, but insisting that neither of us get involved with the other discipline.
Thank you. I think that was a very nice and balanced answer that we have here. I'd request Professor Pal for some concluding remarks.
Bruce, thank you very much. I mean, it's not much that I have to say. I've had a wonderful talk. I think I just come in to give you thanks for coming here. And of course, thanks to our team for connecting. I understand there's a little bit of a technical glitch for which you're delayed by 5 minutes. But coming in very early in the morning, I think at your time, I must thank you particularly. But I should say that the point that you mentioned about Zoom or any of those platforms like Google Meet, connecting us all over the world is very relevant. And I think all of us have been extremely busy, actually, I must say. Initially I thought we have less work, but now we are very busy in giving talks all over the world. So I think this has really created a great opportunity. I mean, that's the fact that without much of travel, we are able to address different sections of scientists or researchers all across the world. So I'm very thankful, however, that we have been able to spend this time. And it's a wonderful talk coming in from your role in the National Academy of Science, your role in the Science magazine, and of course, your great experience of science education over the years. And I think I'm very particularly pleased that you mentioned about the scientific temper. And I think it's extremely important. I mean, however you call it, to have a scientific breadth of mind, scientific bend of mind, I would say. And I think it's extremely important for cultivating this from the school level. So I think you have really touched upon the extremely important points. And of course, apart from the fact that the biology of the virus, coronavirus, I'm myself amazed to see the length of the virus that you showed. I mean, it's really great, and the kind of things the kind of research that people are engaged in today in combating the coronavirus. I think it was a great talk from the standpoint of the biology. And of course, for a non-biologist, it has also been a great experience of learning biology and learning more about science education and the scientific research, how to do scientific research. It was a great talk. Thank you, Bruce, again, for addressing us in the Institute Colloquium. Thank
Well,
you.
thank you. Thank you very much. As you heard, I've learned a lot from India, so I thank you, all you too.
Yeah, I see that. Yeah, you have been to India multiple times. Yeah,
But yes.
and even,
And we
I know
hope you're
also
going
MS
to come back
Swaminathan.
and visit us eventually to our new
Yes,
campus sometime, Bruce.
I hope you can make a physical visit to our campus
Okay.
sometime.
Okay. Thank you very much.
Thank you very much. Bye-bye.
So thank you everyone for listening and joining us here. Thank you, Professor Pal. Thanks especially to our technical support team who've been up here beyond office hours helping with the live transmission. And of course, thanks a lot again, Bruce. Have a good day. And we hope to welcome you back to India again sometime. Thank you.
Thank you very much, Andy. Bye-bye.
Bye-bye.